Good morning. I want to start by apologizing for my inability to join you physically this morning, but I've sent in this video to explain, to discuss with you my project, my research, which focuses on the discovery, the targets, mode of action, and potential therapeutic roles of novel anti-diabetic agents. My name is Dr. Pauluojo. Uh, you'll recall that when I came to your class the other time, I talked that there's an increasing incidence of type 2 diabetes, and that is partly caused by the fact that uh, drugs that are currently available for the treatment of disease, many of them have side effects, some of them and are very expensive, and most importantly, there's no drug that can restore normal glucose metabolism. Also, none of the drug is able to prevent the development of diabetic complications, and that's an issue uh, that is very paramount to us now. So these factors made it important for us to begin to look for glands and amphibian skin secretions and other sources for agents that we could develop into novel drugs for treatment of type 2 diabetes. You also recall that I mentioned that there are two forms of diabetes. There's type 1 diabetes, in which you inject insulin, and there's also type 2 diabetes, uh, in which case the problem is either the body is not able to produce insulin sufficiently, or that the insulin that is being produced cannot be used properly. And so in that case, what we do to tackle particularly the problem of beta cell dysfunction uh, resulting from inability of the body to secrete sufficient insulin is that we look for agents that could actually improve insulin secretion, either by interacting with beta cell receptors or by working through known pathways of insulin secretion, that's the KATP or the PKC-PKA pathway, or agents that could stimulate the release of other beta cell hormones such as GLP-1 and GIP, two hormones that are also known to augment insulin secretion within the body. And we are also interested in seeing what happens at the molecular level by studying the changes in the expression of key genes that are involved in this in insulin secretion. Again, for insulin sensitivity studies, insulin action, we also interested in agents that could improve the glucose uptake or improve insulin sensitivity. That's how the body reacts to injection of insulin, and this is actually very important. The inhibition of fat deposition, particularly since we know that obesity is a risk factor of type 2 diabetes. So any agent that is able to promote weight loss will be good. Will have a significant effect on type 2 diabetes. And so again, we also want to see glucose, what happens to glucose tolerance and gene expression, the expression of genes that are involved in insulin action. Generally, my activities are divided into four categories. One of them is the discovery and target and actions of novel agents that I've shared with you. But I'm also interested in drug discovery and design. And so over the past few years, I've been using in silico tools to design drugs by combining peptides from exogenous and endogenous sources, and then again this is to generate compounds that we could test in the lab. And also we want to look at I'm also interested in the role of endocrine disrupting chemicals in the type 2 diabetes. I'm going to talk more a little about this shortly. And I'm also particularly interested in the regulation of non-classical beta cell receptors. Now, for my work on plants, the whole idea, I've identified isolated compounds from a number of plants, from amphibian skin secretion, and also, as I said to you earlier, we're also developing some of these compounds. We're developing hybrids of them, and to see what their effect will be in the treatment of the disease. And of course, interest is also in understanding the site and the mode of action of this compound, with an overall aim of trying to develop something that will have potential therapeutic role in correcting insulin secretion and insulin action in diabetes. These are some of the plants that I've worked on before. The ones in these cycles here are the ones that we've been able to characterize phytochemicals from, and the ones in the box over here are other compounds that we're currently working on, or that we've worked on briefly in the time past. But over the past few years, my effort has been directed mainly into working on peptides from amphibian skin secretion. The paper we discussed with you the other time in class was based on the peptide Tigran 1R that was actually isolated from this frog here. In all, we've isolated about 200 different peptides, and we've about 68 of them have we've been able to characterize their insulinotropic actions. And so some of these peptides now we're currently working on them. We're developing hybrids of them to improve their either their stability or to make them less toxic. Some of them that we discovered to be toxic, and also to increase their potential of being taken by a pharmaceutical company for development. Do you wonder why we work on amphibian skin secretion? The thing is that amphibians do not have any defense mechanism, and so they are endowed with this gland within their skin that produces a slimy compound, slimy secretion. That slimy secretion is for one to help them to escape predators, or it also contains some chemical agents that are that can serve like toxin to wade away, their predators, or also some compounds that could help them in wound healing. And so scientists that have been curious and have been studying that some of these compounds have reported a number of biological activities for compounds isolated from amphibian skin, and we are particularly interested in the insulin releasing action of some of these peptide isolated. And our plan of investigation is usually like this: when we get the amphibian, we get them agitated either by electrical shock or by injection of epinephrine, and the secretion that is produced we clean up using cephalocolumn. Then we fractionate the secretion by HPLC, and we test the effect of each of the fractions one by one to identify the ones that are potent and they are not toxic. When we identify this, we move further to do some more purification, and then we use mass spec to determine what the molecular weight of the peptide will be, and we do Edman degradation to sequence to know the exact sequence of the peptide that is that we have, and the amino acid sequence. And that's the sequence that we send to a company that produce the synthetic replicate of that peptide for us, and we now the powder form of that will be produced, and that's what we use to do more intensive study to know what the dose dependent effect will be, the mechanistic actions, before we finally inject them into animals to know what the acute in vivo effect and the long-term effect of such new drug will be. And I talked to you a little also that I'm interested in working on endocrine disrupting chemicals. These are some of the chemicals that are found in daily products that we use, such as metals and personal care, metals that we're exposed to maybe from petroleum products, or personal care products such as lipsticks and creams, and even pesticide and herbicide that are used in agricultural practices, pharmaceutical drugs. Many of these generate chemicals that are foreign to our body, and studies have shown that some of those can actually elicit reactions within the beta cells, leading to development of type 2 diabetes. So these are some of the other things that we are trying to study, and in the future it's an exciting research area that I'm looking forward to work on. And so this is my research, and some of you you're welcome to talk with me, to have a chat with me. My office is in Stratford in Waterlane campus, AE 439 is my office. You can just come to my office and discuss with me, and I'll be happy to answer questions. Thank you very much.